Welcome to this little video we've put together on work we've been doing on Parkinson's during this lockdown period. I think you'll find as you listen to these talks that people have been extremely busy looking at different aspects of Parkinson's disease. So I hope you enjoy the videos, and at the end of this we're going to have a question and answer session with some of the questions that we come up between ourselves, as well as questions which have been sent in from you, the community.
Hi, my name is Marta, and I'm a PhD student in the group studying the role of gastrointestinal dysfunction in Parkinson's disease. There's increasing evidence suggesting an association between gut problems and Parkinson's disease onset. There are case studies reporting alpha-synuclein accumulation in the gut of patients up to 20 years before diagnosis. Constipation is a risk factor for Parkinson's disease onset, and the gut microbiota, the microorganisms that live inside the patient's gut, seems to be different from those of controls. And this has received a lot of scientific interest recently. But what if you already have PD? Could gut problems affect the disease course? Well, little is known about this. And so during this quarantine, I analyzed a data set from patients who had been assessed from diagnosis up to 8 years. And when I stratified them by constipation severity, I noticed that patients with moderate to severe constipation that is not alleviated by common treatments for constipation, seem to progress faster to balance problems and to memory problems. Now, this is really interesting, and it may hold important clinical implications, but it also raises a lot of questions that we need to answer first. So we've designed a prospective study that hopefully will start after lockdown is over. And we're going to ask patients and controls to complete a set of questionnaires regarding their gut function. We're going to collect some blood samples and some stool samples over the course of 3 years. And we're going to analyze this data together to see whether it can provide us some insights of a mechanism underlying this association between constipation and disease progression. And while we were designing this study, I also noticed that there was no specific questionnaire looking at gut problems in Parkinson's disease. And so just before lockdown, I met with a few groups of Parkinson patients and spouses, and I asked them, what are the relevant gut problems that you've experienced in the past or are experiencing now, and what is the best way to ask about them in a questionnaire? And so with their feedback and after reviewing the literature, I've developed the gastrointestinal dysfunction scale for Parkinson's disease, and I mailed it out to a lot of our patients. And I'm currently reviewing the data and analyzing the data too. Before I go, I just wanted to say thank you for all the time and effort of our patients. It's much, much appreciated. And above all, it matters. So thank you.
Hello everyone, my name is Antonina, or some of you might know me as Nina, and I'm a postdoctoral research associate working with Caroline Williams-Gray. So my main focus of my research is to try to better understand the role of the immune system in Parkinson's disease. As you probably already know, Parkinson's is a very diverse disease, and it might look very different from person to person. For example, we see some people in the clinic that come with more severe symptoms, and these might include memory and thinking problems. And these symptoms can progress quite fast compared to some other people which seem to be having a more benign type of the disease. So I'm trying to understand whether the immune system plays a role in those differences. So over the last couple of years, I've been working on a study which we call the NET-PDD. And this study is trying to answer two main questions. The first question is, do some people with Parkinson's disease have a more active immune system? And the second question is, does this increased immune system activation contribute to a faster disease progression and development of memory problems? So right before the lockdown, we had managed to finish recruitment of all of our participants in the study, which includes 40 healthy individuals and 40 people with Parkinson's disease. And we've done a lot of clinical assessments, including memory tasks, movement assessments to all of our participants. And we also collect a blood sample and a spinal fluid sample. And the reason we do this is because we want to compare whether there are any differences in immune cell populations and also in other markers of inflammation. At the same time, we're also doing PET brain imaging. We're using a scanner which looks very much like this one in the picture. And this type of scan helps us to visualize immune cell activation in the brain. So right before the lockdown, we had almost managed to finish all of our scans. We have about 8 scans left to do once we reopen. So over the last few months, one of my main tasks was to look at all this data that we have collected and compare between healthy individuals and people with Parkinson's disease. And here you see some of our pictures from the PET brain imaging. So on the top panel, you see pictures from the brain from a healthy individual, and on the bottom panel are some pictures from the brain of a person with Parkinson's disease. And the more red color you see, the more active the immune system is in those areas. So at the moment, we're just waiting, hoping to be able to bring our final participants back and finish our scanning so we can have all of our data and compare between healthy individuals and people with Parkinson's. And the aim is to finish this baseline part of the study, and then in 3 years' time, bring everyone back and repeat and see how they've progressed over 3 years.
Hello, I'm Julia. I'm one of the doctors working in the Brain Repair Centre. My work builds on the work of others in our group, looking at the importance of the immune system in Parkinson's disease. I'm helping Dr. Caroline Williams-Gray set up a trial of a drug called azathioprine, which suppresses the immune system in Parkinson's. We hope that this medication will slow down the progression of Parkinson's disease. And I'm sure you know, at the moment we don't have any treatments which can do this. So let me tell you a bit about this trial. Firstly, azathioprine is a drug which has been around for a long time. So we know a lot about how safe it is and what it does to the body. But we don't know the effects that it has on Parkinson's disease. So that's what we're really interested in finding out. We're going to recruit people who've come in through the Parkinson's Research Clinic because we think that a certain type of people with Parkinson's will do best on this drug. So we need to know a bit about our participants before we recruit them. So it will be based on some of the scores that we've gathered from the clinic. We're looking for 60 participants, 30 of whom will take the drug azathioprine and 30 of whom will take a dummy drug or a placebo. And the trial is designed so that neither the participants nor the trial team will know which medication everyone is on. And we'll take that medication for a year. And during that year, we'll monitor closely what's happening to your Parkinson's disease. So we'll do examinations, we'll take blood tests, and there are a couple of optional parts, including MRI scan with PET imaging and a lumbar puncture. And we're particularly interested in these tests because we'll learn a lot about what is happening to the immune system on this azathioprine. With the COVID pandemic at the moment, we've had to hold recruitment. And my time during the pandemic has been spent trying to make sure that things are as safe as possible for our participants when we do open. We're hoping that early next year we'll be able to open the trial to recruitment. And if you are eligible and have been seen in our clinic, then you'll receive a letter through the post inviting you to attend for a visit. Thank you.
Hi everyone, my name is Benjamin. I'm a postdoc in Professor Barker's lab, and I will talk about our work on the selective vulnerability of brain cells to Lewy pathology. As you know, Lewy pathology is a major hallmark of Parkinson's disease. It corresponds to the presence of insoluble structures within cells called Lewy bodies. They are made of a protein called alpha-synuclein, which has aggregated and become toxic. Lewy pathology affects the neurons of the brain in a very selective and time-dependent manner. Certain neurons develop Lewy bodies before others, and some never do. That's why we talk about selective neuronal vulnerability. Our goal is to understand why certain neurons are more likely than others to develop Lewy bodies. Solving this question will help better understand the initiation and progression of Parkinson's disease. We model Lewy pathology using neurons grown in a dish. First, we add an abnormal form of alpha-synuclein to the cells. It acts as a seed that triggers the conversion of alpha-synuclein within cells into aggregated and toxic forms. Some neurons will develop Lewy body-like structures and others will not. And we can detect these structures in the recipient cells with a tracer that emits green light. Maha, a first-year PhD student in our group, uses this model to follow the progression of Lewy pathology over time. With live cell imaging techniques, she can observe in real time the neurons which develop Lewy bodies and those which do not. During the lockdown, Maha used her time to refine her protocol. She will return to work with additional imaging tools to identify the cell subtypes that are most likely to develop the pathology. To talk about my work, we need a little background on molecular biology. In our DNA, each gene contains a code that is interpreted by our cells to produce a particular protein. In addition, our cells can use a special mechanism called alternative splicing to produce different forms of a protein from the same gene. This mechanism also applies to alpha-synuclein proteins that are produced by our neurons. They can exist in 4 different forms, which all differ by their length and by their biological properties. In Parkinson's disease, we know that alpha-synuclein becomes toxic and aggregates to form Lewy bodies. So, I try to determine if certain forms of alpha-synuclein make neurons more likely to develop Lewy bodies. Before the lockdown, I measured their production in many biological samples, in cells exposed to abnormal alpha-synuclein and in brain tissues kindly donated by healthy participants and patients with PD. Since then, I have used my time at home to analyze the results of this experiment. Thank you.
Hi everyone, my name is Sam and I'm a research assistant in Roger Barker's research group. I've been working on, during the lockdown, basically been continuing working on 2 of the Parkinson's disease research studies which I'm involved in, that I help Roger to run. The first is a study which is a sort of 2-part study which is in now the follow-up stage, which is called TransEuro. Some of you probably have heard of this because it's been around for a while now. Obviously, in all the studies, but it is the case in the TransEuro study as well, that we haven't been able to see the patients in person at the John Vangi Centre for Brain Repair as we normally would. So we've been doing what we can to keep in contact with people via telephone, making sure that we're up to date with general things that are important, things like medication and whether there's been any health concerns that we should be aware of, just to make sure we keep on top of things. And something that I've been doing for the TransEuro project, which has been a project in the pipeline for a while, is moving the data to a new database, which is quite a big job because there's lots of data that's been collected over the years. So I've been looking at a lot of numbers and writing code and to move the data into the right format to get it into a new home, basically. And that's a great thing to be able to do at home. As you can see, working from home is something that's easily achievable remotely. So that's been something that's been positive. Another study that I've been continuing to work on, which I worked on before, is the OXB102 gene therapy trial, which is sometimes called Sunrise PD. and is sponsored by Axovant Gene Therapies. We obviously, again, haven't been able to see our current patients. We have 4 current patients on the trial in Cambridge, and that's, of course, because of concerns to do with the lockdown and seeing people in person. So again, we've been keeping in contact with people as much as we can via telephone and doing things remotely, like sending out questionnaires for them to return to us. Of course, the bulk of the main assessments we haven't been able to do, but a telephone consultation is sufficient for the doctors to be able to be aware of any problems that might be arising and to recommend particular strategies which we might help with those things that patients might have concerns with. In terms of the OXB trial specifically, this trial is also in the recruitment stage. So we have 4 current patients, but we're also ideally looking to recruit new patients at some point soon. And of course, we don't really know when we will be able to do this. And the priority for Addenbrooke's Hospital is the safety of its patients and staff, of course. And we're just sort of emerging out of the lockdown at the moment. So it's not quite clear when we will be able to start recruiting again, and obviously it massively depends on the resources of Addenbrooke's Hospital and the various departments on which we work with. So for the moment, we don't quite know when it might be that the trial will restart its recruitment of new individuals, but we do know that that will happen at some point, and when it does, we'll be looking to recruit a small number of patients that will be going into the high dose cohort, which is it's a dose escalation study. So we've completed the low dose and we've completed the medium dose. The final dose cohort is the highest dose. So of this product OXB102, the gene therapy. So that's something to look forward to on the horizon for the research group.
Hi there, my name's Danielle and I'm a research manager. I work with both Roger Barker and Caroline Williams-Gray. I've worked with them both now for over 10 years. And during the lockdown period, I've been working on a project called STEM-PD. Now, STEM-PD is a first-in-human clinical trial which is going to treat patients with Parkinson's disease. During the lockdown period, myself, Roger Barker and Emma Cuttin, who is a senior trials coordinator, have been working really hard on this. And we've been working together to write the protocol, to design the trial and meet with the regulatory authorities to ensure that we've got everything in place for the trial to start. We don't anticipate that the trial will start until the end of 2021 because it takes a long time to go down the regulatory pathway. We really look forward to seeing you all back at the Brain Repair Centre soon and hope that you're all keeping well. Thanks.
Hello, my name is Venkat Pisupati. I work in Professor Barker's lab focusing on developing a stem cell based therapy for Parkinson's disease. I am based at the Cambridge Stem Cell Institute. Stem cells are specialized cells. Under specific conditions, they can be converted into various cell types. In Parkinson's disease, dopamine-producing neurons are lost in a region called substantia nigra in the ventral midbrain. The aim of cell-based therapy is to repopulate the lost neurons using stem cell-derived neural cells so that dopamine is produced at the right location in appropriate quantities. In a collaboration with researchers at the Lund University, we have carefully selected a stem cell line and developed a method to generate dopamine-producing neural cells. Using a number of assays, we have characterized these cells and have shown that they have the right properties. We have a number of projects that are going on in the lab using these cells. One of them is to look at the immunological properties of the stem cell derived neural cells. The patients receiving these as transplant will see the cells as foreign and will mount an immunological response. So it is important to know what is the immunogenic properties of the cells that we are deriving so that we can design appropriate immunosuppressive treatments for the patients getting these transplants. This work is being carried out by a PhD student in the lab, Shamma Qarin. Another project that we are looking at is to use mathematical modeling to look at the various factors that will affect the viability of the cells when transplanted. Another project revolves around the influence of culture conditions on the genetic stability of the stem cells as well as stem cell derived neural cells. The information that we gather from these projects will ultimately be helpful in making a stem cell based cell therapy a success in Parkinson's disease.
I'm Sophie, I'm a PhD student in Roger's group, and I'm going to be talking about how we use stem cells to model Parkinson's disease. So one of the main features of Parkinson's disease is the presence of these inclusions, which we call Lewy bodies, and these seem to be present within affected neurons. And while these are a part of Parkinson's disease, we don't exactly know what the role of Lewy bodies are in Parkinson's disease. So to further understand our knowledge of Lewy bodies, we have to be able to model them, and we do this using stem cells. So we take stem cells and we differentiate them to produce the type of neuron that seems most vulnerable in Parkinson's disease. When we have these neurons, we can stimulate them to generate these Lewy bodies within them. And we can then use these as a way of assessing the role of Lewy pathology, understanding the mechanisms and how it affects the cells. And we can also use this as a platform to look at how effective new drugs that we've identified for Parkinson's disease might be.
My name is Caroline Williams-Gray. I run the group alongside Roger Barker. I just wanted to say thank you very much for watching so far. I hope you found our collection of video summaries interesting. The coronavirus pandemic has, of course, had an impact on all of our research studies because we haven't been able to access our labs and clinics. But I hope these short video clips have demonstrated that we have continued to make significant progress with our research in spite of the lockdown. Our research building is now in the process of reopening, so we're just beginning to start experimental work again in the labs with appropriate safety measures and social distancing, of course. Unfortunately, though, we're not yet able to restart our Parkinson's research clinic or any of our studies which require participants to physically come into the building. We're doing detailed risk assessments around all of this, and we are planning a phased restart of our clinic-based research in the coming weeks. But in the meantime, we are keen to do remote assessments for as many of our studies as possible using video calls. Now, these assessments are going to be really important for us to ensure that we can continue with our aim of mapping out the course of Parkinson's disease over time. As you probably know from reading our newsletters and the information on our website, this strategy of following the course of Parkinson's over time in large numbers of people has told us a huge amount about different types of Parkinson's and how they evolve at different rates. And we're now using that information to help us move towards developing different therapies for particular groups of people with Parkinson's. So if you are overdue a follow-up visit in the Parkinson's Research Clinic or a visit for one of our other Parkinson's studies, we will be in touch to invite you to take part in a remote assessment. Now, in order to be able to do this, you need to have a computer with a microphone and a camera, or you can use a tablet or a smartphone, and of course you need a reasonably good internet connection. We'll explain everything to you though when we call to book in the appointment. We'll then set up the video call and we will email you a link. You just have to then click on the link at the time of the appointment, and that will connect you either to myself or Marta or whoever's doing the assessment with you. On the video call, we'll be able to go through questions with you as we normally would if we were seeing you in person. We'll be able to do some tests looking at your movement, and we'll be able to do some of the thinking and memory tests that we normally do in clinic. We will also be sending out some questionnaires by the post for you to fill in and then send back to us. And we hope it won't be too long before we can invite you back to the clinic and see you in person. So thanks again for watching. And we're now going to move on to a question and answer session where the whole team's going to be involved, and we will try and answer some of the queries that you've been sending in to us over the past couple of weeks. Thanks.
Well, that was fascinating listening to all those videos about what everyone's been up to and the exciting breadth of research we've been doing. But obviously it's been a fairly major problem, the COVID pandemic. So Caroline, how much has it really impacted on the running of the research clinic, which you've been doing for several years, and the trials which come out of that?
Well, it's definitely had a really big impact on us. I mean, the research buildings, of course, have been closed down completely. We have just very recently reopened, but only 25% of the staff can be in the building at any one time, and we still can't bring participants into the building. We're managing to do some work via remote video call assessments. So, I mean, that's allowing us to collect some data, But it means that we unfortunately can't start seeing new patients, and we can't get going with new clinical trials that we were planning for the moment. It's not all bad news. I mean, there have been some positives to the lockdown. It's given us the opportunity to spend time analysing some of the data from our PD research clinic and from some of our other clinical research studies. And we've been drafting a number of research papers that we're preparing now for publication. And in terms of the sort of clinical trials, we were planning a clinical trial of azathioprine, which should have started in March. We now have this period where we're suspended, but that's given us time to step back and think a bit about some of the experiments that we're going to do with samples from the trial. So hopefully when we do eventually get going, we'll be able to do an even better job, and we'll be able to measure various other things that we hadn't really considered before. So hopefully we'll have a better trial at the end of all this.
And in terms of just, the new world, if you like, where we're doing things more remotely, I mean, have you started doing any of that yet in terms of, because I know the clinics have, the building's only just opened. I mean, have you had any experience with that yet, and sort of looking into the future, how that will develop? So
yes, we've started that over the past week or so. So we ran our first PD research clinic the other day remotely, and it went reasonably well, I think. The difficulty is, of course, that normally when people come and see us in the clinic, we would collect blood samples from them, and they're quite important to our research because we want to look at various genetic markers and other markers in the blood. And we obviously can't do that when we're not bringing people in person. But we can collect quite a bit of data remotely. Certainly it's quite a good way to collect neuropsychological data, so data on memory and thinking tests. And we'll carry on doing that for all of those people who we think are going to be at particularly high risk from COVID. So when we do get going with the face-to-face clinics again, we'll very carefully screen everyone according to whether we think it's safe for them to come in.
Great. So Marta, I mean, a lot of your work is obviously very patient orientated. So you really need to be able to work with the patient. So, I mean, how have you found it during this period of time, trying to keep your research going? And obviously you've been very heavily involved with the patient community, trying to get them more engaged with the research. Not that we need to get them engaged, but, get more involved with the research. So how is that working in terms of your research and reaching out and having communication with the patient community?
Well, our virtual door is always open, and during this lockdown, we've been in contact with several patients that reached out to us via Facebook, Twitter, phone calls, and even emails, asking different questions. Just when is clinic going to be opened? Do you have any news about this clinical trial on PD that I was interested in following up? To where can I donate my brain to research? And so we've been helping out clarifying these issues. And just this Wednesday, I had a meeting because we're organizing a meeting with a group of patients, because we're organizing an online seminar series, which hopefully will be done sometime in August. So we have been keeping busy. In terms of research, just like Caroline said, I took the opportunity to just analyze some of the retrospective data that we had, and I wrote a manuscript that hopefully will be coming out that will perhaps feed into other clinical trials that could be followed up after this research.
And I'm right in thinking, Marta, a lot of the questions which we're sort of discussing now have actually come from the patient community. They've been very active at sending in questions relating to some of the work we do here, as well as broader questions.
That's right. I think most of the questions that we're going to discuss here were provided by our patients.
Yeah. So Nina, one of the questions which always comes up and has come up in the questions sent in to us is about causes of Parkinson's disease. So obviously we have a limited amount of time, so we can't go through all the possible causes, but it would be useful just to hear your thoughts on this around the work that you've been doing in inflammation, and whether you think this is still, all these years since you started the research, a critical player in Parkinson's disease, driving the course, or at least causing some forms of it.
Right. Thank you, Roger. So in terms of causes of Parkinson's, of course, we know that the actual symptoms are caused because of the dopaminergic neurons in the brain are starting to die. So these are the brain cells that are producing this chemical called dopamine. and we are still not completely, it's still not completely clear why this is happening, but we think that this is a combination between environmental factors and also our genes. So for example, researchers have identified a lot of genes that slight variations of those might increase your risk of developing the disease. They don't directly cause the disease, but might influence the likelihood of developing it. And some research from Caroline from a few years ago identified that one of the key components of the immune system, variations of that component can increase your likelihood of getting Parkinson's disease. So this was one of the initial signs that we identified linking the immune system with the disease. And the other thing that we also know is in Parkinson's, there's this kind of abnormal protein that starts to accumulate in the brain, which is called alpha-synuclein. So research recently has shown that this protein can directly activate immune cells, both in our brains and also elsewhere in the body. And these immune cells start to basically trigger an inflammatory response. So what we are thinking is that basically this inflammatory response can make the disease worse, can impact the brain cells, leading to further cell death in the brain. So the idea is if we can reduce this inflammatory response, then we might be able to slow down the disease, how fast the disease progresses.
And do you think that's true of everyone with Parkinson's disease, or do you think that's true for just some people? I mean, how widespread do you think this problem is?
Yeah, so this is a really good question. And it leads back to a study that we've done a few years ago, which shows that not everyone is the same in how active their immune system is. So people who, there's a group of patients that seem to be having a more inflammatory response to this abnormal protein. And there's another group of patients that seem to be, their immune system seems to be more dormant. So we think that we shouldn't be treating everyone in the same way. And basically these anti-inflammatory therapies that we're considering might be much more beneficial to a subgroup of patients. And we think that this group is the one actually that goes on to develop dementia and has a sort of more aggressive type of the disease.
Thanks very much. I mean, Caroline, this is a subject dear to your heart. You spent many years studying this. I mean, this heterogeneity of Parkinson's disease, different patients following different courses presenting in different ways. I mean, Nina implies this is all down to inflammation, citing work that you've done. Is that the whole story, that this is really how you behave in Parkinson's disease depends how you respond to the protein in terms of immune function, or are there other factors you think which might be important?
Well, we think the immune system might be part of it, but undoubtedly there are other things going on as well. I mean, Parkinson's is an incredibly complex disease, and we've found not only variations in immune genes that affect how the disease progresses, but also variations in all sorts of other genes. So variations in the gene that codes for a protein called tau, variations in the gene that encodes an enzyme that's involved with breaking down dopamine, variations in the alpha-synuclein gene. All sorts of genetic variations seem to affect the course of the disease. So it's undoubtedly a sort of very complex picture. We've been doing this work, as you said, for a long time now, and our studies over the past sort of 20 years or so show that there is this huge variability in the rate at which the disease progresses. And our research has, if we're sort of putting it all together, it's really enabled us to get a fairly clear picture of which genetic abnormalities and which clinical factors that we can measure at the time of diagnosis tell us how someone is going to do over time. And I think that's going to be really, really helpful, that ability to predict how someone will do over time in terms of selecting patients to put into clinical trials of particular sorts of therapies.
So you would say that there are sort of intrinsic problems perhaps to do with the nerve cells, as well as this inflammatory response and age, and these combination of factors determine how people present and how they progress. That would be a sort of general view on it.
Yes, I think so. I mean, I think it would be too oversimplistic to say, it's all to do with the immune system, or it's all to do with problems with dopamine nerve cells, or it's all to do with accumulation of the tau protein. I mean, we think all of these things play a role to different extents in different individuals. And as we learn more and more about this, I think we'll get better at deciding which factors are most important for a particular patient. And ultimately, what we hope is that we'll be able to target therapies according to that sort of information.
So, I mean, it's clear that Parkinson's disease, seems to have a problem within the nerve cells. But one of the curious things about the condition is not all nerve cells seem to be affected in the same way. We obviously have some of the nerve cells, as Nina and Caroline were saying, dopamine cells, which take a heavy burden of disease. We have cells on the outside of the brain, the cortex. Some of those take have a major problem in Parkinson's disease. But there are other nerve cells which seem completely impervious to the disease process. So, Sophie, I mean, you've been trying to study Parkinson's disease in the dish, which would seem very hard to understand how you can condense or reduce a patient down to a few cells in dish. So how have you been trying to study Parkinson's disease in the dish? And then I'm going to come to Benjamin and ask him how he's also been trying to answer the same question in a different way.
So to understand Parkinson's disease, we have to be able to generate neurons that have Parkinson's disease-like phenotype. And then we have to be able to compare these to healthy neurons and see what the difference is between them. We can do this in various different cell types. So a lot of people use animal cells and animal neurons to look at it. The way that I approach this is to use human stem cells, and we can differentiate these to produce dopaminergic neurons, which are the type of neurons that seem to be affected most in Parkinson's disease. In order to generate the kind of PD pathology in these cells, we can either mutate them so that they can have genetic mutations that have been linked to PD, but because most cases of PD aren't linked to a certain gene, we can also use different ways to induce Parkinson's pathology in these. We can treat these with chemicals that have been linked to Parkinson's disease, like certain pesticides. The way I approach this is to generate this thing called Lewy pathology, which is kind of what Nina was just talking about, which is where we see the presence of these aggregated forms of the protein alpha-synuclein. And we can generate that by treating these cells with aggregated forms of this protein, and then we generate Lewy pathology and we can assess what that does to a cell.
And what's the point of that? I mean, I could say, well, that's great. You can mimic what you see, but how do you take that information further? I mean, what do you do with that piece of information?
So we can look at how the cells respond to this, whether or not they seem to have specific deficits that we associate with Parkinson's disease. And we can really look into these mechanisms of pathology, and we can try and exploit these by using drugs to try and rescue these certain deficits that we see.
So this could be used as a platform for screening for drugs, which could then move into clinical trials.
Yeah, so that's the purpose of it.
Great. So, but Benjamin, I mean, you're looking at something similar, and I think the question that you're particularly interested in is, why is it some nerve cells, like the cells we've been hearing about, the dopamine cells, are very badly affected by Parkinson's disease, but other nerve cells don't seem to be troubled at all? So how have you been approaching this particular problem in Parkinson's disease?
As you know, one of the main features of Parkinson's disease is the presence of these aggregated proteins within the neurons, which are called Lewy bodies. They are mainly constituted of a particular protein called alpha-synuclein. So I decided to adopt an approach that focuses on alpha-synuclein to see if this protein itself could confer a specific vulnerability to some neurons compared to other neurons. And I adopted this approach because alpha-synuclein, like all protein, can possess different modifications that modify its function and its structure. And among these modifications, we have a mechanism called alternative splicing that generate different forms of the same protein, alpha-synuclein, but with different size. And the idea of my work is to determine if a modification in the amount of each of these forms of alpha-synuclein could participate in the selective vulnerability of some neurons compared to others.
And so this splicing, this would be something that different cells do in different ways. So every cell would be exposed to alpha-synuclein. It's just some of them splice it into more toxic forms. Is that the general sort of?
Yes.
Yeah. Okay. And how much evidence do you have for that at the moment? Is that something that you think this is definitely happening, or there's a suggestion that this is the case?
This is definitely happening. I am currently working on two types of biological models. First, fetal brain cells isolated from human embryo, and human postmortem tissue. And I'm conducting an analysis of the expression of this isoform of alpha-synuclein in these two types of tissue. And I can see that in pathological condition, there is a clear modification in the respective amount of each of alpha-synuclein isoform. So there is something changing, but the question remains: what is the feasible pathological relevance of these changes?
Okay, great. I mean, that sounds very interesting, and it fits in a bit with the work that Nina's done previously, looking at post-mortem tissue, just to show the value that looking at the brains of people who've passed away with Parkinson's disease gives us huge amounts of useful information, which we can then feed back into our research. Now, obviously, Sophie's been working with stem cell-derived dopamine cells as a way of trying to model disease and screen for diseases. But, you know, one of the things that we've been particularly interested in, as many as you will know for many years, is whether we can actually try and repair the brain in Parkinson's disease by putting back nerve cells which are missing, in particular the dopamine nerve cells. So this is something we've spent many years working on, particularly with our colleagues in Lund in Sweden. And Venkat, you've been heavily involved with this project, and I guess it would be helpful to sort of, from your point of view, what are the two major areas you think we've sort of been we could usefully contribute in this? I know we've been looking to some extent about worries with what happens to cells with genes, genetic variants, so cells becoming unstable when you grow them, and then also something about the immune response, because obviously the cells we will put into people's brains will not be from the patient themselves. So how far have we got to understanding how safe these cells are and how well the immune system will be able to ignore them?
Yeah, thank you, Roger. Yes, one of the main issues is growing these cells in a dish. As you start growing them outside the body, they have the tendency to acquire lots of changes because of the pressures they experience being in a dish. So these are called the selection pressures that make them to change their behavior. So what we are trying to understand is what are the pressures that a cell experiences in a dish, and how to minimize the deleterious effect of these pressures on maintaining the integrity of the cell. So this is a project that is ongoing, and we have used a novel approach of using sequence-based, looking at the genomic sequence of the cells exposed to different conditions.
So can I just interrupt you? I mean, you know, obviously, I suppose we don't want too many technical details, but fundamentally, obviously, there may be concern that what we're talking about here is that these cells are a little bit unstable and could turn into dangerous cells if we use them in trials, which we'll come on to discuss in a minute. But your impression has been that these cells are actually quite safe. They may acquire genetic changes, but they at the moment don't seem to be bad for the cells.
No. So the longer they are in culture, the more changes that they acquire.
Yeah.
So what we are doing for our from a clinical trial point of view is we are taking the cells at a very early stage, and we are also evaluating their stability using different types of assays to know that they are having the required stability and they don't have any mutations that could have a deleterious effect. So we'll be testing them.
Okay, and those cells, just coming back to the other question I was interested in at the beginning, is are they very immunogenic? In other words, if I was to put them into someone's brain, would the immune system go crazy to try and reject them, or are they actually quite easy to hide from the immune system?
Yeah, so this is another aspect that is important before we go on to the clinical trials is to look at how immunogenic these are, because these are the cells that are going to go into a patient, and the patient will see these cells as foreign. So they will elicit an immune response against the cells. So what we are looking here is how immunogenic these are. And we are employing a number of assays to look at this particular aspect. From our preliminary understanding, or the results that we obtain suggest that these are less immunogenic, and they don't seem to be eliciting the kind of response that would be more alarming. So they don't seem to be that immunogenic at the moment.
And if we were to use immune suppressing drugs, which we will do in trials, that doesn't adversely affect the cells. In fact, it might be a good thing for Parkinson's from what we've heard already from Nina and Caroline.
Yes, definitely. So we're also looking at the impact of these immunosuppressive drugs and how it will affect these cells when it is transplanted. So far, we see that the cells are not affected by any of these immunosuppressive drugs. So it will be beneficial when the patients receive these cells as a transplant.
Great. Well, thanks very much. I mean, so it looks very promising with these cells. So Danielle, I mean, obviously this is a project which you've been involved with for about a decade now, where we have the Transuro project, and now we've moved on with this stem cell project. So one of the questions, I would say probably the most popular question I get asked in clinic, as I did this morning at least 5 or 6 times, when can I have my stem cell therapy for my Parkinson's? So where are we with the trial that we've been talking about for so many years, and when do you see it coming likely to start?
That's a very good question, which everyone wants to know the answer to. So at the moment, we're currently writing the protocol for the STEM-PD study. It's a huge regulatory pathway that we have to go through because it's classed as an advanced therapy and also a device trial. There's quite a lot of paperwork that we actually have to go through. It's not actually that straightforward, and we are hoping to get the trial up and running by probably September next year, which may seem quite a long time away, but actually the hoops that we have to jump through take quite a considerable amount of time, and all the regulatory. So we've got a meeting with the MHRA in August, which will help us pave the way for what we actually need to do.
The MHRA are the people we have to get approval from in the UK, is that right?
Yes, so they're the people that if you want to use a drug or a cell in the human patient, then we have to actually get approval from those people to do that for a clinical trial. So hopefully we'll have some more answers in August as to what we can and can't do, and then we'll be able to take this forward further. Like I say, we're aiming for September next year, and the process is going really well, and actually being in lockdown has helped because it's helped us focus and have lots of meetings, which we probably wouldn't have had time to do. So we're actually moving forward at quite a quick pace.
So COVID's been helpful for this trial?
I think it has actually, yeah. It's enabled us to move forward. Yeah, it's been a bad thing because actually we're not at the point where we're recruiting patients, so it's not been a bad thing.
And I'm right in thinking it's complicated not only because we're having to use a stem cell that we've already talked about, but because we also have to work out how to deliver it to the brain, which again is something that hasn't necessarily been worked out before.
Yes, that's right. And also I think this actually could be one of the first actually in the UK to be done. So actually an advanced therapy plus device. So we could be one of the first actually to do this ever. So it's quite an exciting prospect really. But like I say, there's a lot involved in it. And it's not an easy process, but we're working hard to get there.
Sav, you're involved with a new therapy, a gene therapy and a device system. Now, Danielle's been telling us that COVID's been good for the STEM PD. So for your trial with the gene therapy, has COVID been good for you?
Thanks, Roger. I mean, obviously, the trial, I'm just one part of a huge team working on the trial. But I think, in fairness, you would probably have to say that coronavirus has not been helpful for the OXB trial. And that's just because of some of the things that Caroline mentioned already.
So the OXB is the gene therapy trial.
Sorry, yeah. So we call the gene therapy trial OXB, and that's because that's the name of the product, the thing that's injected, the drug that's injected. It's not, coronavirus hasn't been particularly helpful just for the same reasons that it hasn't been helpful in any way for anybody to be able to see each other. So obviously to keep track of how the current patients we have are, we need to be able to see them. As Caroline mentioned, there are some things we can do remotely, and we're doing as much as we can remotely, but There are certain things which are not possible to do remotely, things like particular types of scans, or taking blood samples, or some tests which require a doctor or a research assistant like myself to physically be there, to be able to do some kind of physical movement or something like that with the patient. So some of those things have been limited, but we obviously have done what we can to collect the information that we need to with the current patients. It's looking like we're emerging out of lockdown now, so soon we will be able to start seeing our patients, hopefully. And in terms of, I mean, we have 4 patients in Cambridge, and because this is a phase 1 trial, which basically, for people who don't know what that means, it basically means it's the first time in which these people have received this very specific thing, so that drug in that format. It means that we have to be very, we have to keep a very watchful eye on the patients and make sure we're closely aware of what's going on. So we will be seeing them soon. In terms of new patients, we don't really know when we will be able to recruit new patients into the trial. That is something that will be being done at some point, but we obviously have to wait until Addenbrooke's Hospital is in a ready state to be able to let us do that.
Thanks very much, Sam. I thought I would just, based on that, just give a brief comment on the GDNF, because various people asked me about GDNF, which is this growth factor, which was the subject of a trial in Bristol and was BBC in a paper on it last year. Lots of people want to know where that's going. I mean, we did hold a meeting last summer in the United States where we've just published it in the Journal of Parkinson's Disease, which was a sort of summary of the field and where we see it going. There's a similar study to GDNF, or a similar factor to GDNF, called CDNF, which is subject of a trial in Scandinavia at the moment, which had a press release earlier this year showing it was well tolerated and safe. At the moment, I mean, there are trials which are ongoing, or well, they were ongoing till COVID. They're currently suspended around growth factors and Parkinson's. So this is an area that people are still looking at. It's not something we look at here ourselves, but it is something that is being looked at other sites. So I don't want people to give up on growth factors. It's just at the moment we're not doing it, but other sites are just re-exploring that data. So I would like to thank every panel this afternoon for doing all this. I would like to thank Charlene for being behind the videoing of all of this. I thought I'd just end by asking Caroline how she sees things going in the next few weeks to months. You've already touched upon it. We can expect people to start getting calls from us to come back, not just remote assessments, but actually coming to clinic.
Yes, we hope so. I mean, at the moment, it is just the remote assessments that we're doing. So people will be getting phone calls from Molly, our clinic administrator, asking them to whether they want to participate in those. We're hoping that in August, we'll be able to start inviting people back into the building. But as I think I mentioned, we will have to screen people quite carefully to make sure they're not at unduly high risk from COVID. So we will be going through that process and just making sure that, we're not putting people at too much risk. But hopefully from August, we'll get some people back into the building, and then it will be a staged approach. So as time goes on and the risk level from COVID drops, we'll be able to get more and more of our participants back. And I would hope by the autumn, we'll be getting back towards something like normal, but we'll see how it goes.
Thank you very much. And I'd like to thank also all the people who sent in questions via the different routes to us. Thank you for all your support in the community, both in terms of the questions you send us, the patience you show us in terms of answering all of Marta's questionnaires, for example, for coming to clinic. Thank you for the interest, and thank you for listening this afternoon.